Welcome to our review on culturing microorganisms. When we're thinking about this process of culturing microorganisms, there's a very clear reason to do this, which is if we actually want to study any bacteria, we need to be able to culture them in the lab. So basically, we're going to grow them in large numbers to see the colony as a whole, as you can see in the petri dish at the bottom. In order to actually culture these microorganisms in the lab, we've got to provide them with everything they need to survive within that stuff we're growing them on. And what we actually do is we use something called a culture medium. Now, that culture medium is going to contain everything those microorganisms need to survive. So it's got a carbohydrate source, which is going to provide them with the energy that they need. It's going to have any of the minerals that they require for growth. It will also have some kind of a nitrogen source, which then gives them any of the nitrogen they need to make the proteins. We can actually have two versions of this culture medium. It could either be a nutrient broth solution, on the left there you can see it, or a solid agar jelly on the right there. What we actually find then is that when we're growing these bacteria on the agar plate, so the solid jelly on the right, then we can grow them in two different ways. We could either select to grow them to form visible colonies, so we could select individual colonies there, or we could grow it so that we get this entire coverage with a lawn of bacteria across the entire surface of the dish. And depending on what it is we're aiming to do with those bacteria will determine which medium we use and which technique we then use. So once we've actually got our microorganisms on our agar or in our nutrient broth solution, then we need to ensure that they've got the right conditions for growth. And in order to do that, they need two key things. One, oxygen. Two, some kind of warmth. What we must remember though is that we're not going to incubate these cultures at anything above 25 degrees Celsius within a school lab. And the really simple reason for that is that if we incubate them above 25 degrees, then we're likely to see the growth of human pathogens. Because what we find is that any of those pathogens that are designed to infect us as a human being generally like body temperatures like ours, so 37. So we keep it below 25 to ensure that those harmful pathogens are less likely to grow. When it comes to actually making these agar plates, then you probably never had to do this in school. Generally what happens is you turn up to your lesson and the science technicians in your school have already prepared them, so you just go and pick up these pre-made little plates. You do, however, need to be aware of the process that's gone into making those. So what we end up with is this hot agar jelly first of all, which is a lovely liquid. You then pour that into the petri dish and then you put the lid back on and leave it to cool, and then it sets into the solid agar jelly that you're familiar with. Once we've actually got those nice cool set plates, we can then transfer our bacteria to it. We've got two options for how we're going to do this depending on what it is that we want to achieve with our bacteria. So if we want to produce individual colonies, then we're going to transfer our bacteria to the agar plate using something called a sterile inoculating loop. So basically it's a wire loop that you can then take the colonies and spread them across the surface of the plate, and you'll end up with individual colonies. The other option is one that you are more likely to have done in school, which is creating a lawn of bacteria. So what we do there is we take a sterile dropping pipette and put a solution containing our bacteria onto the surface of our agar plate. And then we use a plastic spreader, which is kind of like an L-shaped bit of plastic, to then spread it across the entire surface. The idea being it gives us this nice equal coverage across the entire surface, hence it's called a lawn of bacteria. One of the key considerations we need to have in our minds when we're doing any studies with microorganisms is how we can ensure that all we're growing is what we intended to, and not some random other bacteria that's happened to get on there through our poor practice. So what we need to ensure is that we've got an uncontaminated culture. And there's a few key things that we can actually do to achieve this. First one is to use sterile Petri dishes and culture medium. So we sterilise them just by heating them up before we use them to kill any bacteria that may already be present. We'll also be using a sterile inoculating loop if we're using the inoculating loop, and generally we achieve that by passing it through a Bunsen flame just before we use it. Obviously not making sure it heats up and then sizzling the bacteria off the surface of any plates. Third one is that we're going to make sure that our Petri dish has a lid on it, and that should be lightly taped on. Now, you don't seal tape all the way around the edge because that generates anaerobic conditions, and again you're more likely to grow pathogens in that scenario. So it's just a couple of little bits of tape on the edge just to hold the lid on so it doesn't fall off and therefore let air get in, which has bacteria present. Last one is that when we actually store them, then they should always be stored upside down. And that just means that any condensation that forms sits on the lid and doesn't actually come into contact with the agar, and therefore reduces the risk of contamination. One of the required practicals that you need to know about is all to do with culturing these microorganisms. And one of the investigations is looking at how effective antibiotics actually are. So the way that we carry out this experiment then is that we prepare an agar plate with our lawn of bacteria, as we've already described, and then we're going to place these tiny little paper discs which have been soaked in different antibiotics on the surface, as you can see on the right there. One thing we do need to make sure we also include in our experiment is a control disc, which is one that's been soaked in sterile water and placed on the surface of our agar as well. Once we've placed all our discs on, making sure that they're nicely spaced out so that they're not all on top of each other and there's plenty of room to get accurate results, then we incubate that for 48 hours at 25 degrees Celsius. 48 hours later, we take our plates out and then we're going to measure the zones of inhibition around each disc to give us our results. The reason that we're going to include that control disc that's soaked in the sterile water is just to make sure that any difference we observe is due to the antibiotic being tested and not something weird that's in the paper of the disc itself. And what we can also do is use this exact same technique to test other things like antiseptics or disinfectants, or we may want to have a look and see how different concentrations of antibiotics affect our bacterial growth. So all of those follow the exact same technique, we're just soaking our little discs in the different things. When it comes to working out which antibiotic is best, then we need to calculate something called the zone of inhibition. And quite simply, the zone of inhibition is the area in which the bacteria have been unable to grow. So the larger the zone of inhibition is, then the more effective the antibiotic is. So the bigger zone is the best antibiotic to use. In order to calculate this, then there's 3 key steps. First one is get a ruler out. Hopefully you are going to take one into the exam with you, otherwise you're going to come unstuck. So with your ruler, you're going to measure the diameter very carefully. Don't just throw the ruler on and take a vague measurement. Actually line it up, take the measurement, and then double check it again. Once you've got that, you need to divide it by 2 so that we get the radius, and then we just substitute that value into our formula to find the area of a circle, which hopefully you know from maths is pi r squared. So you're allowed a calculator in every science exam. Plug those numbers into your calculator and write down your final answer. Good practice here is always to write down the stages of your working. Just in case you fail to use your calculator in the exam, you'll at least get some marks for the working, even if your final answer isn't correct. If you just write down an answer and you've put one number in the wrong place, you've got nothing. So always show the stages of your working there. Hopefully at the end of this video you can describe how to carry out these processes to culture microorganisms. You can explain how we can avoid contamination. You can recall the temperature at which we must store these at for incubation to avoid the growth of pathogens. And you can also explain how to calculate the zone of inhibition.